This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 847, recorded on December 28th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody. It's a gray day out there. I'm sure that it's cold also, but I'm inside, and I haven't been outside at all today, so I have no idea. It's probably in the 40s, as I can guess, and just looks like a typical winter day. Hello, everybody.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 34 Fahrenheit, 1 degree Celsius, and both weather apps say it's snowing, but my eyes tell me it is not.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 44 here, 7 Celsius, but cloudy and gray and not especially inspiring outside.
Yeah, that's a good way to put it.
And from Madison, New Jersey. No, not from Madison. Brianne Barker.
Hi. No, currently from upstate New York, where it is 36 degrees Fahrenheit and 2 Celsius, and also very overcast.
Right. We can tell from your new background there with the windows and the white that makes you think it's got snow out there, but it doesn't actually, right?
Right. There are patches on the ground.
How many hours north of New York are you?
It's about a 5-hour drive from Madison here.
Wow, that's a lot.
Oh,
wow.
Plus
And you make it actually in 4 hours because you drive really fast.
4.5.
And you're still in New York State?
I am.
How far are you from the border?
Yeah,
no,
right.
we're still a good hour from the border. I'm a bit north and west of the city.
So you're up near like Oneonta or something.
Or Rochester?
No, so the closest places people might have heard of are Utica and Rome. The name of the town is Camden.
Yeah. All right. Well, this is our last TWiV for 2021. And I say good riddance 2021. It's a tough year, although 2020 was pretty tough also.
Yeah, 2020 was worse, but 2021 was
yeah, because we had vaccines in 2021.
Yeah.
But this will probably drop on January 2nd. Sunday is the 2nd, right? Is that correct?
Yes,
yeah,
that's correct.
But the last recording of the year, the last time I will say all the things that I say at the beginning and the end. And we will do a review of the year next week. As we usually do traditionally, which is funny that most years it's a mixture of different viruses.
I wonder what we'll talk about this year.
Yeah.
Gee.
Well, we did have
we
a lot
talked
of
about some other.
interesting-
Actually, this episode we're going to talk a little bit about.
Yes.
Yeah.
We have had other viruses, and we'll definitely highlight those. And we've had some interesting guests as well.
Yeah.
Oh, yeah, absolutely.
So we should highlight. And I would really like to make a highlight film, right, of the year. That would be fun.
Sure.
Of highlights, but I'm not sure I have the time to do
Be
it.
a ton of work.
Yeah, it'd be a lot of work. I had to pull a lot of hard drives out of storage.
You'd have to sort through hundreds of hours of video.
Hundreds, yeah.
Yeah.
Like 3 a week, but I don't even remember which ones were memorable. I'd have to go and look at them over again. I guess what I could do is go to the YouTube and scrub through quickly, but we'll see. Maybe by July it'll be out, you know. All right, today we have two papers for you. The first is in the Journal of Virology. No, MBio, I'm sorry. I saw ASM and I thought Journal of Virology. MBio.
And that
Lassa,
means it's open access.
It
because
is.
It's in MBio.
Yes.
And today I just paid the bill for publishing a paper in MBio. So next year we'll get published and we'll do it because it's a really interesting finding. But this one is a Lassa fever live attenuated vaccine based on codon-deoptimization of the viral glycoprotein gene. And this comes from the Integrated Research Facility at Fort Detrick. That's the facility where Jens Kuhn's work, and he's there on this paper. And they have this work had to be done in a BSL-4, which they have over there in Fort Detrick. It's also from the University of Rochester and the Scripps Research Institute. Do we have co-firsts on this? Does anyone know?
I don't think so. The interesting thing is, I think that Luis Martinez Sobrido is the senior author, and what clued me in that paper is actually from January 2020 was that he's now at UTMB, but at this point he was still at Rochester.
Right. So first author is Yingyun Kai. We don't think there are co-first authors, so apologies if it turns out there are. The footnote's hidden. And senior author Luis Martinez Sobrido. So
I have an RSS reader that I use to look at papers, and when I see one I like, I star it and it gets saved. And then I go back and I don't even look at the dates when I save them. And this one, oh, that's cool. Let's do that one. But I didn't know what time, but it doesn't matter.
It doesn't matter. It's timeless.
Now, Lassa fever is caused by Lassa virus, and it is partially why I decided to do virology. As I've spoken many times, I read that book, Fever by John Fuller, which Dixon knows well.
Interesting. He does.
Was the doctor at Columbia, John Frame?
Correct.
Who was involved in that first outbreak in Nigeria in the 60s.
And Fort Detrick was involved a little later on also. So they've got a rich history of this as well.
Really? Okay. Now, Lassa fever was first noticed in Africa. I think in Nigeria were the first cases.
In Lassa.
And Lassa, Nigeria, yeah. And it continues to be endemic in Western Africa. Hundreds of thousands of individuals every year are infected, and you wouldn't know it. You never hear about it, right? Many 20% have clinical symptoms and signs, and has a high case fatality rate. I remember talking with someone, and he said in some years, thousands of people may die of Lassa fever. It's a hemorrhagic fever. And what's interesting is that most of the infections are zoonotic spillovers from mice.
That's
very little human-to-human transmission.
I think it's a rat, by the way, Vince.
Mastomus? Mastomus
that's right.
species.
It's a multi-mammate rat.
Okay. Multi-mammate.
So there's an ecological aspect to this too, right? Because in years there are no droughts. That's less and less, of course, in this time. But without a drought, they had a good crop, and with good crops, everybody eats. With not so good crops, only people eat, and the rats have to forage, and that's where they get up the courage to go into the people's houses to get the grain that was harvested. And in doing so, the urine contains the virus. It sounds a little bit like Nipah in some ways, but and then they contaminate the local environment, and then people eat the grain with the urine contaminated Lassa fever virus.
That sounds like hantavirus
transmission.
Yeah,
absolutely right.
So it's transmitted by ingestion and not respiratory. That was going to be my question anyway.
That's a good question, actually.
I think it's respiratory. I don't think it's ingested. Yeah,
but on the way to your mouth, you have to inhale, right?
yeah. But I think when people clean up rodent excreta, it aerosolizes.
You're right.
That sort of thing. So there's no vaccines. We'll actually summarize the vaccine situation later. And only ribavirin has been used, which is not great. Has a lot of side effects. And WHO has Lassa fever on its list of priority diseases.
Immune serum works.
Sorry?
Immune
serum works, yeah. In fact, Penny-Pinio serum was used to save Jordi Casals.
That's right.
A long time ago at Columbia Presbyterian. You should read Fever. I think you can get it now on Amazon. They redid it. I have a copy, an original copy. It's just wonderful.
Now, because this virus is contagious by a respiratory route and is difficult to treat and is potentially highly deadly, it has to be handled in a BSL-4, which is going to come into play in this paper because one of the things they're interested in is finding an easier way to study Lassa virus instead of everybody having to be bubble suited.
Right.
And one
BSL-4 is the highest level.
That is the highest level of containment. You can watch TWiV 200 for a tour inside a BSL-4 facility and see what that entails. And just one other introductory thing, Lassa virus. I'll do. Rich normally does this spiel, right? So it's an enveloped RNA virus with two segments of RNA, and it's ambisense RNA, right? They're plus and minus strand for different genes.
Yeah. Yep. Someone asked me the other day, when you did that documentary in the needle, the BSL-4, what were they working on? I said nothing.
Paperwork. That's why lots and lots of paperwork.
That's right.
That's why we were able to go in, because otherwise we couldn't have. And in fact, when Rich and I went to Galveston in 2019, all we could do was look in the window. So we put a suit on in the suit-up area, but couldn't go in. Now, this paper attempts to make a vaccine, and the approach is something I think we've talked about a long time ago, which is codon-deoptimization. And this was, I believe, first done with poliovirus. Eckhard Wimmer published a paper years ago, and I think we did that paper on TWiV. Basically, as you know, codons that specify amino acids, there can be multiple codons for each amino acid, and organisms tend to have an optimized set of codons for protein synthesis. And so you can de-optimize it. You can take, for example, a capsid for poliovirus, capsid coding region, and de-optimize the codons so they don't do well in, say, mammalian cells. And then you have less protein made, and the virus appears to be attenuated. At least for poliovirus, it was a vaccine candidate.
Yeah.
And you can introduce thousands of mutations into the genome, and that really decreases the likelihood that it's going to revert. So that's good for a vaccine strain.
Right. So the idea is that we're encoding the same amino acids, but we're not using the preferred codons.
Yes. You have the same amino acid, so you're not compromising antigenicity at all, right?
Right.
You're just making less protein. And this has been done with poliovirus, respiratory syncytial virus, foot and mouth disease virus, influenza A virus, and Zika virus.
We also talked about this for dengue in TWiV 351, and the polio and RSV we talked about in TWiV 740. It's also been done for DNA viruses in a letter that was sent to us in TWiV 362.
Right.
And I forgot the dengue one. I didn't realize we did that. Wow.
So the thinking is that because there are thousands of mutations in there, and because it's all about codon usage, this becomes extremely difficult for the virus to get out of, because changing one of those, you could have a point mutation that changes the codon usage to something optimal, but it's not going to have enough impact in the background of all of these de-optimized codons to actually have a selective pressure. And so the stepwise process of getting from the attenuated virus to the unattenuated virus is just too much, whereas older attenuated vaccines that were just selected through passage in different animals and cells and such often have, a dozen or so mutations. And if a couple of those revert, then you get a virulent virus, potentially.
So the idea was to try this with Lassa virus. And this is no mean feat, because it has to be done in a BSL-4 to grow the virus and do the animal experiments. It has to be done in a BSL-4. So, and
it's also an RNA virus, so you've got the tricks of working with that, right?
What kind of tricks did you have in mind?
Is the cDNA infectious?
Right. So you have to use an infectious clone strategy, which they, of course, call reverse genetics.
Reverse genetics.
Yeah.
Oh, yes, of course. Yeah, I think that's de rigueur these days. But yes, you can't just mess with the DNA.
They had some precedent for this, because this had been done for lymphocytic choriomeningitis virus, LCMV, which is very closely related to Lassa. So they had some idea that this could work.
So they decided to do this with the glycoprotein gene of Lassa virus. And they introduced 379 synonymous nucleotide mutations to incorporate the least frequently used codons in mammalian cells. That is 321 of 491 amino acids. So, they use the mutation correctly there, nucleotide mutations, but then the rest of the paper, they blow it. They call them amino acid
mutations.
Right. So to give you those numbers in a different way, they changed about 25%, 26% of the nucleotide sequence and about 65% of the amino acid sequence.
Mm-hmm.
Well, none of the amino acids were changed, but they changed what coded 65%.
The codon.
What coded
Correct.
the amino
Sorry.
acids, right.
Right.
Yeah.
I didn't say that well.
Yeah, and so then when you just produce the protein by itself, you get, they said, 50 to 100-fold lower protein levels in HEC293 cells when you just put the DNA in. And so already you see that changing the codon usage is reducing the amount of protein made. So then they put this deoptimized gene into the virus using a DNA copy of the genome, and they recover infectious virus. And they actually have plaque assays in this paper. They have a picture of a plaque assay. Remember the last paper we did with the had plaque assay.
I'm starting to think some authors might actually be putting in pictures of plaque assays just to try to get on TWiV.
That's right. This is very pretty, actually. And this is no mean feat. This has to be done in a BSL-4.
Yeah.
And there's a lot of liquids involved in staining and fixing and so forth. So it's, I would just tell you, for example, we do plaque assays in my lab. You take the agar overlay off, you stain the monolayer with crystal violet, and then after 10, 15 minutes, we pour off the crystal violet into a large plastic container with a funnel, so it goes in there. You do this in the sink. But inevitably, a few droplets get into the stainless steel sink, and it stains the sink. And then periodically, the safety people come by and say, have you been dumping crystal violet? No, it just, a few drops goes a long way. And so now we have learned to scrub it with ethanol before they come. So they don't, I shouldn't be saying that.
Well, when I was in the lab, we had those epoxy-lined sinks that were completely black, which was much better for that because you just didn't have to worry about it.
That's right. Yeah, you were in the old days.
Yeah.
Anyway, so they do plaques. This virus, they do recover virus with the codon-deoptimized glycoprotein gene. The titer is, after the transfection, the titer is lower, but they can amplify that to reasonable titers. They look at the growth kinetics because you want to make sure that you're not making a big impact on fitness, right? Otherwise, it may not be a good immunogen. And I think they say about an order of magnitude lower final titers. The plaques are smaller. So the nice picture of the plaque assay, they compare the wild type with the codon-deoptimized. 2 millimeters versus 1 millimeter, roughly, plaque diameter. So
one of the beautiful things about plaque assays, you can see phenotypes.
Yeah, you get a lot of information from that, I think. Okay, so what do you do next? Well, you put it into animals, right? And they have two kinds of guinea pigs. They have strain 13 guinea pigs, which I think is a wonderful name. It makes me think of some alien guinea pig.
It should be a movie title.
What was that? Area 13 or something like that? Strain 13.
Well, Area 51 was the reconnaissance base, but
so strain 13 guinea pigs, which they later tell us are hard to get. So then they switch to others.
Right, and the strain 13 are inbred also, and maybe it's hard to breed guinea pigs inbred.
They're highly sensitive to infection.
It's not hard to breed guinea pigs when they're not inbred.
100% lethality following subcutaneous inoculation of 1,000 plaque-forming units. The other guinea pigs are Hartley guinea pigs, only 30% lethality. Well, the guinea pig wouldn't say only, but after inoculation of 1,000 PFU. Actually, 2 times 200,000 PFU you need for that. So they said we decided to look first in strain 13. They inoculate them subcutaneously, 100,000 PFU both. So they use the codon-deoptimized. They use a DNA-derived Lassa, but wild type and sequence. And then they use Lassa virus that had never came from DNA. It came from nature eventually, ultimately. All the animals lived after inoculation with the deoptimized. They didn't develop any signs of disease. all of them inoculated with the wild-type virus had clinical signs of infection. Ruffled, scruffy appearance, respiratory issues, weight loss, temperature, etc. And some of them reached the euthanasia threshold where they sacrificed them.
Right.
So,
So
they
in your
don't
animal
develop
use
hemorrhagic fever. They just develop other signs and symptoms, but not what people develop.
Yeah, I think so. I think that's correct.
when you have this kind of experiment going on in your animal use protocol, you list a certain set of clinical signs that you will monitor, and when they add up to a certain number of points or something, then you need to euthanize them. The interesting thing is they also measure weights, and in the strain the strain 13 mice that got the codon-deoptimized one, there's one guinea pig that had this severe weight loss, and then you look at the footnote and it tells you it's because it gave birth to 2 healthy pups. So lost a lot of weight after it had those pups born.
I wonder if you see the same kind of pathology in the Mastomys species.
Yeah, I don't know.
That's a good question.
It's a good question.
Yeah.
It's probably not easy to breed them, but I do know that, if I remember correctly, when I was out at Rocky Mountain Lab, they were talking about trying to set up a Mastomys colony.
My guess is that they don't get very sick.
Maybe not, yeah.
They may be like bats and rabies, I think.
Yeah, that's right.
Well, I think the mice that carry hantavirus don't get very sick.
That's right.
They don't.
That's
They
right.
And that's part of the problem. I mean, if they just got sick and
is the
died out
problem.
in the wild or something, then we'd never encounter them.
Yeah, it just surprises me that we see so much pathology in a guinea pig and not so much in Mastomys. It's interesting to have the change of pathogenesis in such kind of similar organisms.
Co-evolved.
Yes, the guinea pig's not the natural host, so.
No.
And neither are humans. They both get sick.
This is very true.
No, they couldn't detect viral RNA in the blood or in any tissues. They found antibodies to the virus, but they were not neutralizing. These are the ones inoculated with the codon-deoptimized virus. There's no significant histopathological findings. So they say these demonstrate complete attenuation of this codon-deoptimized virus in strain 13 guinea pigs. The authentic, the Lassa virus derived from DNA, on the other hand, you do find virus in the blood. You can find it in many tissues. You can find, and as some of them died, as we said, you can find lesions in the tissues as well, including pneumonia, liver issues, and other things as well. So quite a distinct, very interesting, right? That virus, the codon-deoptimized virus is quite different.
Well, and it's also so interesting that they've only codon-deoptimized one gene of the virus. And so the rest of the virus is wild type. And part of me wonders what would happen if you codon-deoptimized other viral genes instead of GP? Is this just because of it being glycoprotein?
Yeah.
Or could this be something else? That to me was really interesting, that it was really just, wasn't a codon-deoptimized virus entirely. It was just that one gene.
Yeah. It may be too much loss of fitness. Because I know for polio, they also just did the capsid coding region as well.
It'd be terrific if they could modify the virus so that only the Mastomyces got infected and it couldn't be transmitted to humans at all. They can have it all they want. We don't get it at all.
So you want to make a vaccine for the rodents?
I would like the rodent to get all of the infection and humans to get none of the infection.
Indeed.
Because once it gets into humans, it's highly contagious between humans. They don't need the rats anymore.
No, it's not highly contagious between humans, actually. There's very little.
That's how it was first discovered, though, Vincent.
No, it was not highly contagious. It is minimally. As I said at the top, most of the infections are brand new spillovers from Mastomys.
But the beginning epidemic that triggered this whole study back in 1967 occurred in one little place where the doctors were getting sick as well.
I'm sure if you bring a sick person into a hospital, it's like Ebola virus, right? Where you get transmission in the hospital because they're working right on top of them.
Or
and
SARS-1.
Ebola is the same way. Or SARS-1, exactly right. Yeah.
Right.
But most of the new And so those chains can be short because there's, in hospitals, you're supposed to have infection control.
No, I got it.
Although back then they might not have done that because Penny Pinio was airlifted back to New York.
She was.
She was sick. They just stuck her on a 707 with everybody else.
Well, this is true.
So
But
back
do you
then we
remember?
didn't. I don't remember. I wasn't alive in the 60s.
We're not going there.
But I think the idea is, and in fact, I talked to a guy on a TWiV in Rotterdam a couple of years ago, and he said most of the infections now are spillovers. So there's not a lot of human to human. So it doesn't become a human virus. It's always a zoonotic infection, right? So next they want to see, what if we've immunized the guinea pigs now? We have this virus that appears to be attenuated. So they immunize strain 13 guinea pigs, 100,000 PFU subcutaneously or mock with PBS. And then they wait 30 days. It's a month.
Yes,
and then expose them to 100,000 PFU, again, subcutaneously with Lassa virus, the non-codon-optimized. So all the immunized pigs survive, and all the mock immunized lost weight. They had elevated temperatures. 3 of them died, and 1 developed clinical signs and recovered. So all the vaccinated guinea pigs are protected. They didn't see any viremia. They did see some virus in the blood of mock immunized animals, but not the immunized animals. No lesions in the tissues of immunized animals. And they did, again, find IgG, anti-Lassa IgG, but not neutralizing, to which they say, suggesting that neutralizing antibodies did not play a role in the protection conferred by the vaccine. And this is a single
and suggesting that their clinical trials may be a little more complicated.
Why is that? Because you can't easily measure neutralizing?
You can't line up volunteers and just see if they produce an antibody response and say, well, that's a correlate of immunity, because it may not be.
They may have to do FC
may have to actually
assays,
look at T cells. Yeah.
Or even T cells, yes.
Oh my gosh.
Yes.
The horror.
Indeed. Okay, so that was strain 13 guinea pigs. Then they said, well, we don't have a lot of these strain 13. Limited availability, so they immunized Hartley with this virus, and they exposed them with wild-type virus. Again, they're all protected. No clinical signs of disease. No viremia. And again, the mock immunized do develop viremia and disease. So works in both kinds of guinea pigs.
And in both of these cases, when they're using their control wild-type virus, it's a recombinant one. So they've produced it the same way.
Yep. And then again, they look at antibodies in these Hartleys. They find anti-Lassa, neutralizing in a serum of only 1 of 16 guinea pigs immunized and then exposed, which again, they just copy-pasted the same sentence suggesting that neutralizing do not play an important role. I can see Jens doing that. Don't get mad, Jens. We're just joking. A single inoculation is protecting them. All right, so this is good. Now the question is it genetically stable? So they passage the virus in Vero cells. They infect Vero cells at a very low multiplicity of infection, 0.01. They wait some hours. They collect the supernatant. Then they infect fresh cells, again at a low MOI. So they have to determine the titer in PFU per mil at each passage. And they do that 20 times.
And the significance of going in with a low multiplicity of infection is you put in a little virus on a lot of cells, so the virus is going to grow for multiple rounds in that. We're just trying to grow it for as many generations as we can to see if it eventually regains virulence.
And they do genome sequencing of all of selected passages, virus from selected passages. They do growth kinetics. They all seem to have similar growth kinetics. Peak titer, viral titers the same. And they identified 2 changes, 2 single nucleotide polymorphisms. They don't seem to be doing anything. So they think it's stable in Vero cells, Although I would say maybe you should try something other than kidney.
Yeah, I wondered about that too, because Vero cells have that situation of not being able to secrete interferon, and how does that play into things? And just the choice. It would have been interesting to be in on the conversation when they chose what cells to do that. The other thing that I found about this section was they talk about the two nucleotide polymorphisms that they find, and one of them is in the glycoprotein gene, but they could have so easily told us, was it one that had been codon-deoptimized or not, and was it going back to what it, but I looked it up, and it's an amino acid change that's not codon-deoptimized, if I understand it correctly.
I mean, I don't know if I would suspect you would want to look in some other tissue that may be more relevant to pathogenesis, because they do subcutaneous inoculation, then they find virus, at least in the wild-type virus-infected animals. They find them in a variety of tissues. So they don't mention kidney, but hemorrhagic, so maybe endothelial cells. I don't know. I have a feeling it's not going to revert, but none of these have been in thousands and thousands of people. So who knows, right?
Millions and millions of people.
Yes.
Millions and millions, yeah. So it could be. I just don't know. We'll see. Anyway, that's the data. I think they're very interesting, and I hope that it progresses and they can eventually get into at least a phase 1. We'll talk about that in a moment. But they say in the discussion, other studies have shown that T-cell responses are the best correlates of recovery and protection. And they actually validate that to some extent, because neutralizing antibodies are not, right?
And I will say that my experience with LCMV is in terms of thinking about it as something to study T-cell immunology. LCMV is a very popular tool among immunologists to study T-cell responses.
Yeah. And they do say that, however, you can treat people with antibodies, which was done in the 60s, and it's been done more recently take B cells from patients and isolate the antibodies from them. But they do want to have a vaccine. Yeah. And they say people have tried vaccines based on vaccinia virus vectors, VSV vectors, Mopia virus, yellow fever vectors, and others. So there a lot of approaches are being tested. And in fact, CEPI, the Coalition for Epidemiological Preparedness Innovation, is supporting development of 5 different vaccine platforms, different vectors. But they have not been promising the results. For example, they're saying that this one measles virus, Lassa, did not provide sterile immunity. And no documented evidence supports protective efficacy of either chimp adenovirus or mRNA-based vaccine candidates in animal models.
Didn't you? I interpreted that as maybe they just don't have the data yet. I don't know.
Could be. It says the limitations of these candidates underscore the need for additional platforms. They're sort of positioning their data saying, maybe we need this because nothing else is working. But
you've got to have the My Research Saves the World line in there. Yeah,
yeah, of course.
Well, and the VSV platform, they say it requires high doses. And then, so I immediately thought, well, what if you put in this codon-deoptimized glycoprotein into the VSV platform? Yeah. Maybe that's what they're thinking is, hey guys, try in some of these other platforms, try our codon-deoptimized.
So why insist on a sterile immunity for this? Why?
That's a good question.
Use the same criteria as we're using now for the SARS-CoV-2.
I agree. I don't think you should try to get sterile. That's a high bar, right?
Yeah. Well,
you keep people out of the hospital. That's the main thing.
Well, and especially for a virus that's not widely spread by person-to-person contact.
Yeah.
Yeah,
that's
and
right.
I'm sort of, as I read that statement, I thought, well, how many things do induce sterile immunity?
Yeah.
Another recurring theme.
Yes.
Indeed.
Yeah. Another use for this, while waiting to get to clinical trials, and they point this out in the paper, is, well, right now we've got to go and suit up and go into the BSL-4 to work with Lassa virus. But if this codon-deoptimized thing, is safe, and it's really kind of looking like it probably is, then maybe we can work with it under less containment and do Lassa experiments at a higher speed because we don't have quite all those obstacles.
I just wanted to point out CEPI. I interviewed the CEO of CEPI back in December of 2019, and we'll put a link to that. He talked about the philosophy. It's a nonprofit. They raise money, and then they use the money to support vaccine development, where companies don't want to do that, right? Because for whatever reason, and they're obviously involved in Lassa. They're involved in Nipah vaccines, and of course also they've supported a lot of research on COVID vaccines. So that's an interesting approach, right? To try and get around this problem of for-profits not wanting to make certain vaccines.
Yeah. Well, yeah. I mean, basically vaccines that prevent diseases of poor people, it's going to be very hard to turn a profit on that. So for years, people have been trying to come up with schemes like this, and CEPI is one of the major ones.
Where are they located?
CEPI?
Yeah.
I think they have offices here in New York, because I didn't email him actually when I was arranging it. I emailed his associates, right? I think they're in New York. Let's look.
Yeah, I'm just
They
looking.
might be my neighbor. Maybe they're in this building.
That's right. Wait a minute, they're right next door.
We have offices in Oslo, London, and Washington. Okay.
Oh,
Not New York.
Headquarters are Oslo.
Interesting.
Yeah. Here we
Founders Bill and Melinda Gates Foundation and the Government of India.
Wow.
go. Huh. Richard Hatchett is the CEO, and he is the one I interviewed. And I think he was previously at Duke, if I remember. It's an MD. Yeah, it's really a great idea. And then there's, of course, the nonprofit that's working on antivirals that we interviewed some of the principals a couple of years ago. What is the name of that one? Ralph Barrick and others.
Right.
oh,
I can't remember.
I feel like it was something like Prepare, but
Yeah, that's nonprofit for antivirals. Let's see if that works. No.
Ready, READDI.
Ready, yeah. It worked. It was the fifth hit. There you go. Okay, so it's good stuff. Good stuff. Okay. Thank you for locating those older TWiVs, Kathy.
Mm-hmm.
Now we have a SARS-CoV-2 paper, which is a Nature article. It's an accepted, right? It's accepted and accelerated, so
Yes.
it's been peer-reviewed. It's called SARS-CoV-2 infection in free-ranging white-tailed deer. And we have two co-first authors, Vanessa Hale and Patricia Dennis, and the corresponding author is Andrew Bowman. And this comes from Ohio State University, the College of Veterinary
Ohio
Medicine.
State University.
Sorry, yes, of course. That was
right.
my last trip in 2020. I flew out to Ohio State, got my picture taken, and flew to the football stadium. My host insisted that I had to do that, Cleveland Metro Parks Zoo, the Ohio Wildlife Center, USDA, and the NIH. And one of the authors here is, Linda Safe, who I interviewed in Ohio State. She drove over from the veterinary school, which is like 2 hours away. Actually, did I? Was she on that? No, she was at Kansas City at the coronavirus meeting. Sorry. Anyway, she was the PhD advisor of Steph Langle. Small world. So let's review all the non-human animals that can be infected, apparently, with SARS-CoV-2. We have domestic animals, cats, dogs, ferrets, and we have wildlife under human care, like big cats, Asian small-clawed otters. That was new to me. Western lowland gorillas. That would be your former person there, Kathy.
Nadine Lambersky in the
Thank
San
you.
Diego.
That was a cool one. Mink, of course. And then some free-ranging wildlife, mink in Spain and Utah, and they say which purportedly escaped from a nearby farm. Okay. Then they've done some experimental infections. Hamsters, and these animals can be infected. Hamsters, North American raccoons, striped skunks, white-tailed deer, raccoon dogs, fruit bats, deer mice, domestic European rabbits, bushy-tailed wood rats, tree shrews, and multiple non-human primate species. I love these names like bushy-tailed wood rats. Isn't it great how they name them? Lovely. And then transmission among cats.
Also, based on our most recent TWiV, we now know just regular old
mice,
lab mice.
Yeah,
that's right.
And ACE2 receptor experiments suggest cetaceans could also be infected if that's the barrier.
Yeah, but remember that paper, which we soundly thrashed because they put deer, what was it, mink at the bottom of the list?
Right.
So I'm not a fan of these predictive things. Transmission among cats, ferrets, fruit bats, hamsters, raccoon dogs, deer mice, and white-tailed deer. Vertical transmission in experimentally infected white-tailed deer. And I think we discussed the antibody, the serological study done this year where they found antibodies in 152 free-ranging white-tailed deer sampled across Michigan, Pennsylvania, Illinois, and New York.
Right.
Right. And in the discussion they said...
It's funny.
Anyway, I'm trying to find it. Oh, the many animal health experts hoped the results were an artifact.
Yes.
However, based on today's paper,
this
might just be the tip of the iceberg, as they say.
Yeah, so the argument against the antibody studies is, well, a lot of things infect deer, a lot of things we don't even know about. These could be cross-reactive antibodies against SARS-CoV-2. Maybe it's not really in them. This paper kind of puts the nail in that, I think.
So they sampled 360 free-ranging white-tailed deer from 9 locations in northeastern Ohio. This was done between January and March of 2021. They they used RT-PCR, and they found 35.8% of nasal swabs from white-tailed deer. Okay, so tell me, how do you get a nasal swab?
So that was
from
one
a white-tailed deer?
That was one
I
of
can
the
think
things
of a couple of strategies.
That was actually one of the things that I was really curious about. It was fascinating to read this paper while I was in upstate New York with my dad, who spends a lot of time thinking about white-tailed deer, and we had some great conversations. I learned some really useful things in thinking about this paper, but we actually talked about how one does a nasal swab in white-tailed deer, and I looked into it. So if you look in the methods, this was actually done in an area of very high deer population where deer have to be euthanized. And so this was these nasal swabs were taken from deer that had been euthanized in these study sites. And they say it's part of a deer population management program.
I see.
And so the deer were baited with corn for 2 weeks prior to the culling session. And so I think there are two reasons why that was really interesting to me. One is that this was, if they're in a place where there's a deer population management program, there is a very high concentration of deer. And so as we might be talking about a little bit later, we can think about, say, deer to deer transmission or something like that. And here we're looking at a very concentrated deer population. And also these deer are frequently interacting with corn that humans have put out for them in terms of bait. And so that's one interaction point between deer and humans.
Right. And just a little bit more on the corn as bait. It's not just researchers putting out corn as bait. In many states, it's legal to hunt over bait for deer. But even when it's not, and I think they even mentioned in this paper that in this deer overpopulated area, people are not supposed to feed the deer, but they do extensively, and this is well documented in the wildlife literature, especially in suburban areas, which are often infested with deer. People will put out some corn or a feeder even in the backyard because they want to see deer. And for anybody who's listening who does that, they say, "Oh yeah, we love to see our deer." Stop. Okay, don't do that. Just really, it's horrible for the deer. It makes them sick. It's just not a good idea.
Alan,
but
I'm
yeah,
sure
it does
I'm
attract
sure
a lot of.
just listen to you and stop.
Yeah, exactly.
And not only are there corn baits, there also are salt licks that people will put out. And so you can kind of imagine how that's going to concentrate the deer and possible human interaction opportunities.
Can you find the virus in their droppings though also? Just like you can in humans?
Any virus or this virus?
They didn't look in this paper.
Virus.
They don't report it. It's possible.
Yeah, maybe.
I'm just wondering, does corn make the deer meat taste better?
There's argument about that. So the corn-fed deer on game farms is obviously a thing. But I've the venison I've had was wild shot, so I don't know how.
Does corn make beef taste different?
Absolutely.
It does. Yes, it makes it more tender. And actually, the problems with feeding corn to beef are very similar to the ones of feeding corn to deer. It gives them digestive problems. They'll develop diarrhea. They don't digest it well, but they can't stop eating it. It's like Doritos. They go to the feeder and they keep chowing down on the corn, even though it's not really nourishing them very well.
Yeah,
but it also selects for that highly toxigenic E. coli strain, 0157. So that's why at a lot of animal annual fairs where they have herds of cattle, and they start feeding them corn to not to sweeten them up, but just to keep them well fed. The switch over to the E. coli, which is pathogenic, can sometimes contaminate, let's say, the cake tasting contest or the hot dog eating contest or the et cetera. And that actually happened in New York State once at the New York State Fair.
Yeah, Kathy, my understanding is...
corn ain't so good for nobody, no how. And even if you're going to make ethanol out of it, forget it.
Kathy, my understanding about the corn bait is that it is simply about getting the deer to come close enough to either see them out the window or for hunting purposes.
That's true. I knew a lot of people that loved having apple trees in their backyard as a result of that, because when they would fall off the trees and ferment, the deer came along and had a party all throughout the fall. And that's the basis for a lot of hunts.
I don't put anything out, and the deer are just all over the place.
They like your flowers.
Yeah, in your area.
They eat the grass. And as I said before, they will not eat the Pachysandra for some reason.
Because they know you want to get rid of it.
But they eat the trees. The dogwood trees in particular. I see them jumping up and pulling down the branches.
Oh yeah.
Yeah, it's fine.
So the answer to how they did the nose swabs on the deer is you kill the deer and then you swab the nose.
All right, thank you. So they CT values less than 30, so a good amount of RNA. Prevalence estimates 13.5 to 70% across these 9 sites. And interestingly, male deer and heavier deer are more likely to test positive for SARS-CoV-2 RNA. So do you guys think that baiting, is it more likely that an infected deer would come to bait than a non-infected deer? Are you not concerned about that at all? I'm just wondering. I have no idea because I don't know anything.
I mean, it is striking in this paper how many of the deer were infected. Because PCR testing of deer, you think, oh gosh, that's, you're never going to find that. You got to get an active infection right when it's replicating, and wow, a third of these deer are carrying virus carrying RNA anyway. As we know, that's not necessarily infectious virus, but it's impressive. And I guess there's a possibility that there's a bias.
Yeah. I mean, they say it's unclear whether dating the deer,
not dating the beer.
Baiting the deer contributed to the increased frequency of virus that they found. So it's a caveat.
Yeah, I would wonder
if
they did get the same percentage in a less concentrated area of deer.
Right.
Is there any way to get a swab without killing the deer?
I think so. You just probably dart them and then do the swab and they wake up.
That's
All right, so they're just, you can't just go up and grab their head and swallow.
No.
Right? You
I
gotta
wouldn't
dart
recommend it.
No,
I really wouldn't
cannot.
recommend it.
All right.
I asked that question.
So it's impossible. Yeah, unless they've caught their foot on a fence, then you could swap them, right? All right, so they did genome sequencing of 14 specimens from 6 of the 9 sites. And these were collected, they say, 6 weeks after the peak of Ohio's 2020-2021 winter epidemic of SARS-CoV-2 in humans. So they were dominated by the B.1.2 viruses. And similar viruses were found in deer at 4 sites. They also found a rarer lineage, B.1.582, about 1% of human samples in 2 deer samples at 1 site. They did not find alpha or delta.
But this is because this is before those viruses were identified.
Became widespread,
Greek
yeah.
letters.
Yeah, so they found a total of 3 lineage, the 1.2, the 1.596, and the 1.582.
So how do you suppose the deer are picking it up from people?
Well, they do discuss that, don't they?
Mm-hmm.
They do.
Let's save it. We'll save it.
Okay.
Let's save it till we finish. We're not much more data here, Dixon.
Okay.
They identified B.1.2 at 4 sites, but found no evidence that these were transmitting across deer at the site. So in other words, independent isolations. They think each site experienced a separate human-to-deer transmission event of a slightly different B.1.2, which they could tell by looking at the genome sequence. And they estimate 6 human-to-deer transmission events were observed. It's a lot of transmission events.
Yeah.
They say the largest, so during the human outbreaks, there were 3 co-circulating Ohio clades in humans, and the largest seeded the deer outbreak. They got actually infectious virus from 2 of the samples, which is cool. And they don't mention much about that. They think there was deer-to-deer transmission. They do find a number of amino acid changes that appear to be unique to the deer. They're rare in humans. They're not non-existent in humans, but they are rare. And they talk a lot about those, but they say we can't really distinguish if, in particular, there's one amino acid change, E484D, if it arose in an unsampled human virus and was transmitted to deer, or arose de novo in deer. So this is the problem with the data. They don't know if these changes that they observe arose in deer or simply came from humans and we just haven't picked them up in humans.
I liked how clear they were about that.
Yeah. There's a lack of deer surveillance and there's a lack of human surveillance.
Yes.
They just don't have enough data.
Yeah.
That sounds familiar.
And that's one of the things they say. We need to do more surveillance, not just in deer, but in other. So this could be the establishment of a new animal reservoir for SARS-CoV-2. We need to do more surveillance to find out, not just deer, but all these other animals. I'm not sure it's going to happen, but they would like it to happen, right? I suspect this is going to be infecting many different animals in the wild, right? So here's some numbers, very interesting numbers. 30 million free-ranging white-tailed deer are distributed across urban, suburban, rural environments in the U.S., and they can live at densities of more than 45 deer per square miles. There are 700,000 free-ranging whitetails in Ohio, and there are also 440 commercial deer farms in Ohio. Wow.
Yeah.
And they say, as we have discussed, plenty of forage available around urban and suburban residences that draw deer into close proximity,
to
not
Vincent's
only
house.
with humans, but in my house and also animals, right?
Yeah.
So they say it's unsurprising that deer in urban sites were at high risk for infection.
And they also point out that there's all the human detritus, our trash, the feeders you set out, the bird feeders you set out. You've got, I'm quarantining with COVID. Well, I might as well at least fill the bird feeder and cough on that and set it out, and the deer comes and licks it. And also streams that could be contaminated with human sewage, because we know the virus passes out in human feces.
So, Dixon, that sort of gets at your question, how But then they say, does it transmit? It must transmit among deer. And they say deer are social animals that frequently touch noses.
Yes.
Aw, isn't that cute?
They do.
They frequently
And they also
touch
do
noses.
a lot of snorting.
Yes.
When
Really?
they get upset and they get, threatened, they snort. Or if they encounter something they're unfamiliar with. I remember I used to own a little piece of property up in New York State, and I camped on it for the first time just to see what it would be like to, live part of my life there, of course. And in the morning, before I even got out of the tent, all I heard around the tent were little hoofs, and then I heard it was almost like a and in the fall you can see their breath really goes very far out. All right, so we did a study on this with regards to another virus that we were interested in at the time. I forget which one. I guess it was SARS-CoV-2. How can you protect yourself? How do deer that are in a big herd start to snort? I can just see this as a particle transmission event.
And they're going to be totally anti-mask. They're not going to go for that at all.
Absolutely. I mean,
Yeah. And they also in this study they see more of the males infected than the females. The males fight.
Yes, they do.
They snort and lock horns and bump up against each other. Typical male behavior.
At night when I'm outside, I can hear the male horns clacking against the tree branches.
Oh, interesting.
As they walk around. There's so many of them walking around the woods. We have a couple of acres of woods only, and clack, 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 clack.
In fact, that's a hunter's trick. They have horns that
you
they clack
can get
together
antlers and just
to see if they can draw in a large buck. That sort of thing.
One of the other places that they mention is wildlife hospitals. And I have a friend who works at a wildlife recovery center near Philadelphia. And she says they wear masks all the time.
Right.
So, but it doesn't preclude, like in the interim when an animal is being brought in or something like that.
Yeah.
You were, of course, referring to the people wearing masks, not the deer.
The animals get
on. Yeah, of course.
Yeah.
So they don't know if they get sick. They're wondering if they get sick, but they say in the challenge studies there's been subclinical infections, but the numbers are small, so we don't actually know.
Yeah,
I guess
I looked at those studies, and it looks like there were in total in all of the clinical studies there was perhaps one deer with a slight fever. And some with some eye and nose discharge. So it doesn't seem to be too pathogenic in the deer.
So there you are.
Based on this, my bet is on it spreading widely in deer. I mean, just the numbers of deer they find with it, these can't all be, and they estimate about 6 spillback events occurring. So there's pretty clearly deer-to-deer transmission going on here, and we're never going to run out of deer.
Yeah, and I just wonder how many other animals may also be getting infected and potentially being a way that you could have transmission from human to deer. And so I could imagine, say, deer mice also being an additional source of infection for the deer. As I was talking about this with my dad, his first question was, well, do they also measure the eagles, ravens, and coyotes? Because he said that they are they will be such predators for deer. If anything happens to the deer, they will be immediately beset upon by eagles, ravens, and coyotes. And so he said that would be the next place he would check.
Huh.
Good idea.
And so 30 million deer shows you what the absence of their primary predator species, which is the mountain lion, means for the ecology of the entire woodland area and the peridomestic area as well.
They actually have a few primary predators. There are the cougars, but also wolves.
Right, we got rid of
huge
those,
numbers of deer. We got rid of all those from the east.
What about coyotes? We have coyotes.
We have coyotes. No, not even eastern coyotes will eat adult deer.
Why not?
They're too big.
Cats.
Too big.
Wow. Too big,
and there are too many other dogs and cats to eat.
That's right.
So they've got
so
plenty
many
of food.
dogs, so little time.
Huh.
Yeah, so coyotes, eastern coyotes are bigger than western coyotes, but they still don't prey significantly on deer.
No, they don't.
So we displaced all the predators of the deer, and then now the numbers of hunters are actually dwindling fewer and fewer people are doing that, so we're not really adequately replacing the predators either.
That is true.
So obviously one of the concerns is that the virus establishes itself in deer, changes in some way, then pops back into people, maybe with other consequences. Who knows? So you have to be careful, obviously, not just deer, but other animals. I'm thinking, though, we have a lot of focus on SARS-CoV-2, obviously. Have people looked for other human viruses in wildlife? Probably not that much, right?
I don't think so.
So there is some screening of deer, at least in this part of New York, for chronic wasting disease.
Yes.
But that's about it. But you could imagine that because there are samples being taken to do chronic wasting disease surveillance, in theory, you could do some surveillance more broadly.
Yeah.
Right.
I mean, just deep sequence the nucleic acid and see what's there, right?
And by the way, if somebody wants to do that study, you should call up the veterinary school at Tufts and ask for their wildlife clinic. They've got freezers full of samples geographically coded from all sorts of different wild animal species. It's just in New England, but I'm sure there are analogous institutions elsewhere.
Sorry. Are all the deer going to switch over now to the Omicron strain, do you think?
That's a good question. I would assume so. I assume they switched to Delta at some point, and this is an old sampling, right? So if they are still sampling, we should see that.
Well, there's an open question of how much reinfection you can get. How much immunity do the deer acquire after an infection? Can these deer that got infected with B1.2 now get Omicron, or is it going to only infect the deer that didn't have it already?
The deer should have their own alphabet to name their variants.
Yes.
Well, so if you're a deer in a red state, are you going to be more susceptible than you are in a blue state?
Maybe.
I don't.
If there are higher circulation rates.
If they don't wear masks, Dixon.
But the deer density is actually higher in suburban and urban areas.
That's right.
So,
Exactly.
I have to say, every day, whenever I'm home, I see deer, day, night. The funny thing is, we have this, for the holidays, we put this deer on the front lawn that's lit up, right? And at night it's lit, and you can see deer walking by. You can see the shadow of the deer.
You call that a deer? I've seen
I'm sure they're saying, what the heck is this? What is this? I mean, tons of them. Yeah. We have all families.
Or honey, come over here. Rudolph is back. Come on.
And in the summer, they give birth, and that increases the numbers. And it's just incredible.
It is.
Yeah, there's a lot of them. And then I don't put any food. My neighbor used to put out a salt lick, but he stopped doing that. It doesn't have to. They just come anyway, and he can watch them.
So, but you do have this sort of a triad, even a quadrant now, of infections that are co-shared by deer and people, ehrlichiosis, babesiosis,
Lyme.
Now you've got SARS-CoV-2. What's next?
We are sharing the earth with other animals, Dixon. That's the way it's going to be.
Definitely are.
And there's no reason we can't, right?
That's why New Jersey's car insurance rates are so high, by the way, because there are so many more deer killed.
I don't think that's the primary reason New Jersey's car insurance. I think the people are contributing to that a lot as well.
You think the insurance companies might have something to do with that too? But I think there are more deer killed in New Jersey per square mile than there are in many, many other states, for sure.
Maybe.
But it may be the leader in terms of that.
I have had many deer killed in front of the house.
I'm sure.
Just don't, if you see a deer jump out in front of you, don't veer to avoid it. Just hit the brakes and hit it.
That's right.
And I've also had deer die in the backyard. I think I've told this story. And I drag them out to the front because otherwise the town won't take them. I put them right next to the road.
You move the body.
And I put it on a sled. One of my kids, those plastic things that you go down in snow.
Toboggan.
And I bring it out there. And then one I remember, I put it near the side of the road. And the next day it was gutted by some animal. Had eaten all the soft tissues out of the gut, and what was left. Somebody was hungry. Maybe it was a what would have eaten that? Yeah, there's no. You said coyotes don't eat them.
Well, if it's already dead, a coyote might scavenge.
No, I was told that it was eagles, ravens, coyotes.
Yeah,
birds. Yeah, the birds of prey.
Coyotes. Once the deer is dead or severely injured, then the coyote will go after it. The problem is that they won't go and take down healthy deer who are reproducing. So,
okay, we have some time for email, so let's dive in to our mailbag. Alan, can you take the first one?
Okay, I am diving into the mailbag here. And Ricky writes, "Hi, everyone. I stumbled upon your podcast almost a month ago and really enjoy the insight you offer on this pandemic. I received the J&J vaccine in April and was due for a booster. However, I tested positive for COVID on November 15th. I decided to get monoclonal antibody treatment on the 19th when my lungs started to feel inflamed and irritated, despite having what my doctor called a mild infection thanks to the vaccine. The infusion made a dramatic difference in my recovery. Overall, I'm feeling much better, but I still have night sweats and a slightly elevated resting heart rate in the 80s until I take my beta blocker in the 60s for high blood pressure, which can be uncomfortable. I found quite a bit of literature on cardiac-related issues post-COVID from the Mayo Clinic, Harvard, and other reputable sites. I also found an interesting article that criticized some of the messaging in science, specifically as it relates to this pandemic. I know the panel can be critical of media coverage and wanted to hear your take on how scientific data are released to the public. As you can imagine, it can be very scary for those of us either going through or recovering from COVID. Thank you for your time, and I apologize for the long-winded email. P.S. Keep preaching the good word on vaccinations. I know I am." Ricky, that was not a long-winded email. We get many, many longer ones. And provides a link. Did Ricky sent this link?
Yeah.
Okay. Yeah. To a Stat News story, straightening out some information about cardiac.
Yeah. So
issues.
Early on, they said the virus is destroying your heart, and
right.
So it's about messaging and how we have done this, and you message things before they're really ready to be messaged, right? But we've talked about that a lot. It's driven by preprints, and just pandemic does it, and that's what we try and help with here, to try and back off and take a slow look. But we've talked about, the flawed German report was tweeted by more than 19,000 accounts and covered by nearly 400 news outlets. In the headline, by a cardiologist, COVID-19 is creating a wave of heart disease. Cardiologists, well, that's their field, so.
Yeah.
So I just want to pose the unasked question. He was going to get his booster, but instead got COVID-19 and got monoclonal antibodies. So is the recommendation still to wait after getting the monoclonals before getting a vaccine dose?
When I went and got my booster on Black Friday, there was a question: have you received one of the things? Have you been diagnosed, or have you received monoclonal antibody therapy within the last? I forget what the time frame was.
Okay, so then I think it's 90 days. Then.
Okay.
Yeah, I think that's what Daniel says. Yeah.
Yeah.
Yep.
Okay. So as soon as his 90 days is up, he should
probably get boosted.
Go ahead and get his booster. Yeah.
Kathy, can you take the next one?
Lori writes, "Dear Vincent and company, I wanted to bring this comment to your attention. I'm pretty sure you have seen it, but I have made a habit of rereading it with every apocalyptic virus of concern that comes along. I often think about the following scenario. Imagine we had an influenza pandemic with uncontrolled transmission for 2 years and the fancy scale of genomic surveillance we have today. With hemagglutinin less tolerant to amino acid changes than spike and a higher mutational rate than in coronaviruses, would it be fair to think we would have many more viruses of concern in 2 years than we do now? If so, how would reporting on each fuel the variant-specific mass hysteria, let alone the feasibility to properly characterize their biology or go ahead and tweak or remake vaccines, etc.? My issue is that this kind of data shouldn't be made public, that is, news material. It leads to mass hysteria and nothing else. Nothing changes in terms of non-pharmaceutical interventions and vaccines, but ridiculous outcomes happen, such as stock markets crashing and travel bans being imposed. Sorry about the rant, and all the best, Lori." And she sent a link to a Nature article that I haven't looked at yet. The title is "We Shouldn't Worry When a Virus Mutates During Disease Outbreaks" by Nathan Grubau, Mary Patron, and Eddie Holmes. That was published in February of 2020.
Yes.
Yes.
And was widely disregarded, of course.
Yeah.
I think it's a good question. What would happen in a flu pandemic? Would we? Because now we would be sequencing way more than we have done in the past, and I mean, I'm sure you would find variants emerging. I just don't know if the same amount of coverage would accompany them as this one has.
Apparently, there is something going on in Israel right now with regards to migrating birds, where more than 1,000 cranes coming out of Africa died recently of what they attribute to flu. And they said, watch out for this one because this is a much more lethal variety than we've encountered in the past. So,
If you're a crane.
If you're a crane.
Yes.
Well, there's a spillover there. Right?
Yes, there could be. I think where the analogy breaks down is that flu has been maybe not as much studied in real time with billions of dollars on the table as SARS-CoV-2.
Yeah.
But historically, there's so much information going into it that we have a pretty good grasp on flu viral dynamics, and it comes around every year. And so I don't think it would even be possible to generate this kind of hype around flu variation because there's been so much study on flu variation already.
Really?
I
think about 2009, and I mean, the vaccines that we have for flu at the moment are nowhere near as good as
but that's all known.
Well, it's known by us, but it's not known by the general public.
I just, having sat in rooms with the flu folks who complain about the fact that they cannot get anybody to pay attention to what goes on with flu, it just...
But, okay, so, prior to this pandemic, if you had to pick two families of viruses that were going to be responsible for the next one,
flu.
You might have picked influenza.
Yeah,
right.
Would have been top of my list.
And if the hype went up to the level of, this is as bad or worse than 1918, I think we could be there pretty quickly.
Maybe.
And, and we only have a vaccine that's 60 to whatever % effective and so forth. So,
but I think a lot of what is driving the hype with SARS-CoV-2 is that we knew nothing at all about this virus to start with. So we're starting from zero, whereas a new flu, okay, it's a new flu. 1918 happened. We know kind of the shape of flu pandemics. I think there's a certain familiarity there that might tamp down some of the variant.
I'll grant you that. But also, I would say, yes, this virus came along and we didn't know anything specifically about SARS-CoV-2, but we did know about other SARS viruses,
yes.
MERS, coronaviruses, and we know other things about other, virus infections. So we're not starting from like some totally unknown microbe that we don't even know what it is, So, it would be interesting to see how much of, mass hysteria there would be that would be variant specific.
Yeah, I think the hysteria around a real flu pandemic like 1918 coming around again would be more about how the heck do we fix these vaccines that we haven't been able to fix for 50 years. Whereas with SARS-CoV-2, we've centered on variants, and that's become the story because this virus has played out in this particular way. I just don't know that a flu pandemic would play out in the same way. It would probably just be, it's still there because we only have vaccines that are 60% effective. I don't know.
So when Ron Fouché was on, I guess that was this year, he showed two graphs of variant displacements for influenza virus and SARS-CoV-2. So there are these graphs where the colors of the variant, the variant goes up and then another one displaces it. And you get the same pattern for influenza. So we already know this happens, right, in a seasonal setting for influenza virus. So, and again, I think the scientists know this. Maybe if the public is confronted with this, say, during a flu pandemic, maybe, and I think it's fueled by certain pundits and then the press, right? And they would say, oh, look at all these variants of influenza virus. It's just like the COVID pandemic. I don't know. It's hard to know, but it might be the same issue. I just don't know.
Yeah. Maybe
Brianne,
we don't have to go there and find out.
Yes, I
hopefully not. Yeah. I hope so. Brianne, can you take the next one?
Sure. Sue Ellen writes, just wanted to tell you how much I enjoyed learning all about smallpox on episode 822. I hope you do more of these basic virus history type episodes. They are great and answer a lot of questions I have knocking around in my head. Sue Ellen, you should listen to some of the archives.
Yes.
Well, one that I would like some clarification on. I believe that smallpox stayed pretty stable in terms of its ability to infect throughout its history. So the smallpox vaccination that I received as a baby was the only one I needed. Just like the measles one that kids younger than me got was the only one they ever needed. Yet, with flu, for example, we have new strains every year and new vaccines are needed. Same is true for COVID. At least new variants keep arising, and with each one, we wonder if the existing vaccines will have to be modified. So, why are some viruses so stable in that they don't require new vaccines or new responses by our immune systems, and others change so frequently that we need to keep making new vaccines for them? Sue Ellen was in Roswell, Georgia, where it is currently 57 Fahrenheit under clear skies and heading for the mid-60s today. Weather I miss. I think part of it has to do with the types of vaccines. But I think that there's also a virology side to this, and I'd be interested to hear others' opinions.
I remember we did this measles paper where they mutated the gene for the glycoprotein extensively, and there were parts that could not be changed at all. And the analogy, Alan, I think you were on that, was when the airplanes came back from bombing missions, they would see what parts had bullets in them, and then they would make those. The
parts that hadn't been hit, they would put armor on because
those ones
they
went down
realized those were the bombers that didn't come back.
Yeah. So there's this. So I think some proteins, all viruses vary, right? And some proteins are more flexible than others. So polio vaccine has been the same, measles vaccine, many others, yet, others. And I don't know that we understand that plasticity.
Yeah, I don't know that we understand the plasticity either. I will just note that some of the vaccines you mentioned are live attenuated and may give a somewhat more diverse immune response against more parts of the virus, and maybe a little bit more robust of an immune response. Whereas the other ones you're comparing to are not live attenuated, at least most commonly. But I don't think that can explain all of it.
I have pulled up the slide that I showed during my class. It's actually a concept test looking at the influenza virus and the measles virus. And this is the take-home message of this experiment. Surface proteins of measles cannot tolerate mutations, whereas influenza surface proteins can.
Did you call them mutations?
Yeah, I
did. Busted.
Aha.
Yep, because I'm sure that's how it was discussed in the paper.
Yeah, that's the observation, but we don't know why, right? Why is it tolerant and this one is not?
No,
that's
but it is a difference in the surface proteins.
Yeah, sure.
Yeah, and I think of it kind of like different viral strategies for what works. And there are many ways to be a virus. You can be like herpes virus, where you infect your host once and you just never leave. And you just pop out occasionally to infect new hosts. Or you can be an acute infection, and then you have to deal with the immune system on a short-term basis. And you have to deal with the problem of what happens when you've infected all the susceptible hosts. And in the case of poliovirus, well, by that time, more hosts have been born. And so you infect the susceptible hosts that were just born. In the case of flu, it's the seasonal cycle that we've gone into. And you need a new strain to come around because you don't have the susceptibility. And so the flu viruses have evolved. They've evolved in birds, but they have this ability to shuffle around their proteins and get in that way. Measles didn't do that, and measles at the brink of extinction now because we have an effective vaccine. But before we had an effective vaccine, its solution was to be so ridiculously contagious that it had kind of the polio strategy. Well, there's another host out there somewhere, and we're going to find them.
Dixon, can you take the next one?
Yes, I can. Anna writes, "Hi, Team TWiV. I'm a longtime listener, and I cannot thank you enough for the valuable insights and expert opinion you have provided during this pandemic. I'm a molecular biologist by training and longtime cancer researcher in that field. Although immunology and virology are not my main disciplines, I am able to soak in the literature and all that you discuss on here to translate it to the best of my ability to friends and family. I am constantly referencing your podcast and papers you cite to inform others, so thank you. I have two questions for you, the first being very relevant to this moment. In the case of a new variant, like Omicron, can you identify the experiments that can be done in the laboratory immediately to understand whether vaccine-induced antibodies can neutralize this new variant? I don't quite understand how these experiments work and why they haven't already been done and reported in the case of Omicron. I know that human data will come, however, that takes time, and as we know, rather. These in vitro experiments should be, in theory, done much faster, but I would love to understand how those are done and what the limiting time constraints on this is. Second, and the last question is unrelated to COVID, but one I have always been curious about and never fully understood. Do you have a simple explanation for why we were never able to develop a vaccine for HIV? From what I loosely understand, HIV somehow evades the immune system, therefore a vaccine would be tough to develop. And yet, you have had guests on your show who are working on HIV vaccines, which means that scientists still believe it's possible. Are there any other viruses similar to HIV in this sense? What makes it so different than other viruses that we were unable to develop a vaccine against? Thank you so much for any insights to these questions you might have. What you do here is such a value to the community, especially during these times. Thank you for all your fantastic scientific discourse and for making it enjoyable to listen to. Anna. That was a complicated.
Go ahead. Hey, Brienne, to answer the HIV question.
Yeah.
Okay, so for the HIV question, HIV is a virus that can integrate its genome as provirus into an infected cell and lead to this long-term latent infection. We can't identify the cells that are latently infected and remove them. And so basically, once we are at the point of someone having a latent infection, there's not really much we can do. And there are some reasons why it's awfully hard to eliminate cells with a latent infection. That plays into the vaccine problem because with many vaccines, you could have a vaccine that blocks, say, 80% of viruses from getting into cells. You have a small amount of virus that still gets into cells, but that's not enough to make you sick, to send you to the hospital. And your immune system kind of clears up that remaining little bit. It's okay that the vaccine is not perfectly sterilizing. With HIV, if there's even a little bit of virus that makes it around the vaccine-induced immune system, then you're going to start one of those latent reservoirs, and the virus is going to sort of get the upper hand to be able to reproduce for a long period of time. So in my mind, the biggest piece of this is that you would need to be able to completely sterilize with a vaccine right away in order to stop the formation of that latent reservoir. There are also some ways that we can talk about HIV's variation and some of the difficulties in biochemically working with some of the proteins involved in terms of making an immunogen. I think those problems are really solvable. And I think the biggest problem is about making an immune response that can block that formation of a latent reservoir, or coming up with some way that we could ever get rid of a latent reservoir once one was generated.
And a major problem in HIV is we don't know the correlates of immunity. There are a few patients scattered around the world who have managed to, not progress and who've controlled the virus. But we don't have examples of, this person was exposed and is now immune to the virus. because everybody who was exposed has the virus still in their system. So that's kind of the starting point for a lot of vaccines. Well, what are the correlates of immunity? Let's induce those. Well, we can't get past step one. So who's going to tackle the first question?
So when the letter was written, must have been very close to the first detection of Omicron, but the neutralization assays started to come out that first week, right? After Thanksgiving. And I would say 1 to 2 weeks is what it takes. You have to have either authentic SARS-CoV-2 virus, in which case you would need a BSL-3 lab, or a pseudotype, a VSV or a lentivirus pseudotype, which you could work with in a BSL-2. And you'd take, for the vaccine question, you would have stored samples from the vaccine trials from people who participated, and you could pull those out of the freezer, mix the serum with the virus, and then measure infectivity. So it doesn't take a long time, but it does take time. And I think a week is amazing that you can get all that and coming out, but they're not certainly peer reviewed yet.
There are some that are now peer reviewed that are out.
Right.
So some people apparently can avoid peer review. Yeah. So I know a certain individual who happens to be in my institution who sends papers to Nature and they are published the next morning, apparently without peer review. And so I don't understand why some have an exception, but they can, and they're neutralization assays, like we just said, and they can go pretty fast.
Yeah, the issue with those, of course, is even if the science is all done perfectly and the results line up, the conclusions are very limited, or they should be very limited. You cannot know vaccine effectiveness in humans from an experiment in a dish. So all you can say is the serum did or didn't neutralize the virus. We don't know if that affects vaccine effectiveness, and that for that you have to wait for enough people to get infected who are vaccinated and unvaccinated. And then you find out, as we now know, the vaccine still provides extremely robust control of symptomatic and serious infection.
I mean, that's part of this communication issue, right?
Yes.
So these experiments were done and they say, oh, the sera don't neutralize well. There's, especially only after 2 doses, 3 is better. But people get scared. And then it turns out, hey, most of the infections seem to be mild so far. Maybe that's because of immunity.
Yeah.
So people get very scared, and I don't think this is productive. I mentioned this to Daniel last week, and he said the wrong people are getting scared. The unvaccinated people should be getting scared. Anyway, as our letter writer said, it's a real communication issue. All right, we have 2 short ones. Let me wrap with those up. Rudolph writes, please help me. I'm fully vaccinated, including a booster. Every time I go to a store and see unmasked people, I get quite irritated. Should I change my attitude? Please advise. Thank you. So today I was on the train, New Jersey Transit. You need to wear a mask, and there's no eating because
you need to wear a mask.
Yeah. So the lady across the aisle from me was drinking coffee and didn't wear a mask. So I thought, I'm not saying it. I'm not going to say it. I don't want to get in a fight. So did I get irritated? I said, no, I'm fine. I'm triple vaccinated. What do I care? So I don't think you should get irritated. That's not a productive. And believe me, I know about irritation, and it's not productive.
Believe me, he knows about irritation.
Grumpy for nothing.
Yeah, that's right. Grumpy Vinny. So you shouldn't get irritated. I wouldn't say anything because they might hurt you.
Yeah.
You are good to go. Keep your mask on. That's my advice. What do you think? If you want,
be sure you're wearing a good mask. They're, increasingly saying don't just wear a cloth mask. Wear some kind of N95, KN95, KF94, something that's more than a cloth
the study
mask.
You covered a few weeks ago, was it Bangladesh, where we finally got controlled data on masks, and the cloth, random bits of cloth essentially did nothing, and the paper masks didn't exactly do what all the mask proponents would like you to think that they would do, but they did have some measurable effect.
Yeah. Yep. And finally, Martin writes, I have really enjoyed listening to the back catalog and the current podcast. A question that came up among a group of co-authors was, what happens when we run out of Greek letters for designating variants of concern? Martin is at the University of North Carolina, Greensboro. I don't know. Go back to the beginning and put a -1 on it. I don't know. What do you think,
there
guys?
are other alphabets, Hebrew, Cyrillic,
Yeah.
Lots of letters that have other names.
So for influenza, they just give them an isolate name. They don't give them Greek names.
They could do like hurricanes and just come up with a list of human names.
But Vincent, the influenza names do have the initial place of isolation still associated with the name, and we're supposedly trying to get away from that.
Exactly right. Different culture.
Mm-hmm.
I think by the time we get to Omega, we're going to be caring a bit less about the variant of the week. That's just my prediction.
You think so, Alan?
Yes.
The news cycle will not let it die.
But you know what? I'm going to hold on to pi for as long as I can. That is going to be so much fun.
Yeah.
That's pi coming next.
How many letters do we have left?
Brienne?
10, I think.
Brienne's frozen.
10?
10? Okay.
10 left? Okay. Okay, folks, let's do some picks. Our last picks for 2021. Dixon, what do you have for us?
Well, one of my all-time heroes died over the last few days. He didn't die over the last few days. A few days ago, E.O. Wilson died. He was 92. I had the rare privilege of introducing him to a lecture he was giving at Columbia. I actually chose him as the lecturer, and then I had the privilege of sitting in my office with him for about 20 minutes before the lecture actually started. I had 6 of his books, and he signed them all. And I had this big book by him called Ants, and he wrote that, co-authored that with Bert Holdobler, which is a remarkable book in many respects. It's both encyclopedic, but it's also very readable. It's not a tome about this ant and then this ant, and there are stories behind each one of these ants. And he was a marvelous biology storyteller, just like David Attenborough. When he signed it, though, he doesn't sign it E.O. Wilson only. He draws a little picture of an ant next to his name. And it was very charming to see such a great man give homage to such a small creature, because that little bitty tiny creature enabled him to see the world through the lens of multiple species of ants, and it's almost as varietal as human culture. And he developed hypotheses based on what he saw in ant behavior as to how human behavior might also mimic some aspects to that. The next reference that I picked, the conversation, was centered around that concept, because he drew quite a lot of flack from a lot of biologists by saying, what a horrible way to generalize from such a small, not insignificant, but certainly regimented by chemicals, because each caste of ants within a colony is behavior driven by the chemical signals it receives. And we're certainly nowhere near that. We're way beyond anything that would approach that kind of behavior. But nonetheless, he could see a lot of parallels. And he was a good, his ability to draw remarkable generalizations from such limited observations that held true over the years when people doubted them and went out and sought the proof against him and ended up agreeing with him. He was a remarkable individual and suffered a lot from discrimination at Harvard, because I think he overlapped with a lot of the molecular biology movement that happened during the 50s and 60s and 70s. And there was a movement at one point to actually get rid of his biology department because there wasn't room enough for molecular and regular biology on the campus. And he had to fight for his life, basically. And he won, because it turns out you need both of those things. You can't do without the other. They're linked indelibly. And so therefore it's not a political thing, it's a biological thing. You need both classical biology and molecular biology to go hand in hand. And you can see how wonderful that's played out over the years in other fields as well. But just know that when I sat down with him, I knew I was in the presence of greatness. And so I will always feel that way about him.
Yeah.
I went to pick this, and I saw that you had already picked him. But you have more insight into him than I would have, so it's good that you did it. Unrelated to him, though, this website, theconversation.com, there's an interesting note at the bottom by Beth Daly, who's their editor and general manager. It says, "It is easier than ever for special interests to spread disinformation on vital issues. To inform the public about what's really going on, we help experts drill down into the facts. We can't do this without your help. Please support us." And they ask for a donation. I think that's a nice way to put it, right?
Right.
Yeah.
Absolutely.
I'm going to borrow some of that.
Yeah.
I'm sure they'll loan it to you.
Imitation is the greatest form of flattery, right?
This is true.
Brianne, what do you have for us?
So I have an article from Science American that I came across that I really enjoyed. It was called The Best Fun Science Stories of 2021. And it was a link to a group of different stories from all aspects of science that have come up this year. They're just really sort of interesting. You can, for example, read about potty-trained cows, or some evidence on sort of sightings that could be UFOs, or floral mimicry, lemurs being the first primates to have rhythm, or non-human mammals to have rhythm. Just also ways that people were looking at code breaking in old letters. And how to do that. It's just all sorts of really interesting, fun stories to read. And so I love sort of reading and learning about new things. And every time I read one of these, I was like, whoa, cool. So I just thought it was a fun list of stories.
It's a good roundup.
It's nice.
It is.
Cool illustrations, too.
Yes, they are.
Very nice. Kathy, what do you have for us?
Well, first of all, to accompany Brianne's, I just happened to hear on the BBC this morning the story about the Mulu and the cows learning how to be potty trained, and they're calling it the Mulu, and the interview with one of the people involved, and so that was kind of fun. What I had picked was one from University of Virginia, using sound for data analysis, and it talks about a chemical engineer who works on chemotaxis in bacteria, and how she hooked up with somebody who analyzes sound. And so they put in a grant to study the chemotaxis by, could you trace the patterns of where the bacteria go and make musical things out of that. And so there's 2 links there that you can click on to hear what chemotactic bacteria sound like, and bacteria that are not doing chemotaxis. And it's pretty subtle, but I did hear a difference. The first time I did this, when I saw the article, I couldn't tell the difference, but I was in a hurry then. But today I spent more time with it. So just an interesting idea of using sound for data analysis. And they say basically other fields do it a reasonable amount, but it's underused in biology.
Cool.
This reminds me of people who set DNA sequences, make music out of it, right?
Mm-hmm. Definitely.
And they can find patterns that you wouldn't normally see, and so forth. Yep.
Right.
Alan, what do you have for us?
I have a book that I just finished reading recently. It's Owls of the Eastern Ice by Jonathan. I think the last name is probably pronounced Sloft. Jonathan, apologies if I got that wrong and you're listening. It's a So this fellow is a wildlife biologist, which always fascinates me because, my study organism was in a lab and that has its own challenges. But wildlife biology has some rather dramatic stuff to deal with a lot of the time. So he got interested in the Blakiston's fish owl, which is native to a very small area in Japan and the far eastern coast of Russia. And it is the world's largest owl. There are a couple of pictures of them in the book, and they're enormous. They're almost a meter tall. It's like a flying Ewok. It's just this huge brown bird. And they live in these old-growth forests. As I say, the majority of them are in the eastern part of Russia. And they need very specific habitat because they feed on fish and frogs. And a lot of the time, the waterways where they would have their access to fish and frogs are frozen in that part of the world. So they need a stream that stays clear through the winter because it's got a fast enough current, but it also has to have fish in it. And there's a lot going on, with what has to happen in the habitat. But there was also very little understood about these owls because they're very shy of humans. So he went there and started working on them. And he's interacting, of course, not only with the owls, but with the people who live in this region. And it's an extremely remote, rural area of Russia. It's like Alaska, just huge distances through vast forests and little tiny settlements where it's hard to get supplies. And so it's got a great set of adventure stories of interacting with the locals and finding his way to where the owls are and finally figuring out how to trap these owls so that he can tag them. And it's just a ripping good read that gives some wonderful insight into how this type of research is done. And of course, I would have liked to have, read about some of the infectious diseases that might be present in these owls, but he wasn't there to study that. So that'll have to be a subsequent analysis. But really, really cool slice of biology and of culture.
The world's largest owl.
World's largest owl.
Are they endangered?
They are, because this area is heavily logged. And so one of the things that he was there to do was the logging one of the biggest logging companies in Russia was actually pretty forward thinking about the ecology of the place, because I guess they didn't want a lot of bad PR for wiping out a bunch of species. And these owls also are pretty significant to the people locally who they don't exactly revere them necessarily, but they think they're pretty cool, right? Because it's this unique, ginormous owl. And so the lumber company was fine with him figuring out where the habitats were, especially as his data started to indicate that there are very specific parameters. And once you figured out where these owls were, you could build the logging roads elsewhere. And a lot of what the owls need are things like they need a standing dead tree near a stream. And the lumber company doesn't want the standing dead tree. It's not good timber anymore, so they could just leave it there. And so it's a neat human and nature interaction story that also incorporates how to relate to industry. And I think he's with one of the big nonprofit organizations that's active in this type of preservation work as well.
Isn't that great? You can write a book about one kind of owl.
Yes.
Oh, yeah.
Or one kind of anything.
Yeah.
And people will read it. That's great. Very good. I have 2 TWiV picks because we did a paper on Lassa virus today. It reminded me of TWiV number 9.
TWiV number 9.
Recorded on November 22nd, 2008. Dixon and I.
We had a good time.
Fever, the discovery of Lassa virus. Vincent and Dick recall the discovery of Lassa virus in Africa in 1969. A non-fictional account of the story Fever inspired Vincent to become a virologist. Part of the story took place at Columbia Presbyterian Hospital, where both Vincent and Dick are employed. Dick remembers many of the key players. This is when I used to write multiple sentences in the descriptions. And now I just do one.
Yeah. So for people who haven't listened to any of the old episodes, this is even before I joined. So this is Vincent and Dixon, and it's a 40-minute episode of TWiV, just the two of them chatting, and it's a good one. It's old school TWiV.
This was the first year. Yeah, November. We started in September of that year. Yeah,
we didn't have any letters to read.
Right.
We had no letters. There were only 2 links here. We had a podcast pick of the week.
Right.
Because I started by picking podcasts and then it morphed into just picks. So that's one. And then more recently, TWiV 548, which I recorded in Rotterdam at the European Congress of Virology. This was recorded May 2019, I think. No, that's when it was released, but I think it was around then. And we had 1, 2, 3 guests, and Marion Koopmans was my co-host. But it's called Mice, Shrews, and Caterpillars. I couldn't think of anything else because we have one person working on Lassa virus and mice, and another working on shrews, which had Borna virus, and a third working on caterpillars and viruses that alter their behavior. But anyway, the Lassa virus, Stephen Gunther works on Lassa virus, and he's done a lot of genome sequencing to track where these viruses come from and so forth. So that's a good one, if anything, for his part, but it's all good. The Borna virus story is also interesting. We did that story on TWiV where they, I think a couple of German squirrel breeders got Borna virus infection. They died, actually. Yeah. They got it from the squirrels, and it turns out they're actually in shrews, and the shrews give it to the squirrels. Anyway, there's a lot of TWiVs out there, and they're very cool. You should check them out. We have 2 listener picks. One is from Larry, who calls us TWiVniks. You've pretty much replaced Netflix for me. And his pick is an XKCD. I think that Brianne probably picked last week or the week before, right, Brianne?
I think so,
yeah.
Yes.
Yes.
It's about
Still
immunity.
Good.
It's immunity. It's very good. And Larry says, place your cursor over the strip to see the punchline. Yes. And Jeff writes, looking for information about the potential impact of the Omicron variant, I ran across this website by the National Institute for Communicable Diseases, which is the National Public Health Institute of South Africa. There's a very nice dashboard which gives a summary of hospitalizations and mortality due to SARS-CoV-2. So that's a slow loading page, at least.
It's got a lot on it. It's very, very detailed.
There you go. Weekly respiratory pathogens, daily hospital surveillance, weekly reports. Very good. Yeah. Very nice. And nice graphs, too. Thank you. Jeff is at a company called VaxCyte, VaxCyte. Nice name.
Yeah.
Mm-hmm.
All right. That is it for TWiV number 847, our last of 2021. You can find the show notes at microbe.tv/TWiV. You could send your questions, comments, picks of the week to TWiV at microbe.tv. And throughout December and January, if you donate to parasiteswithoutborders.com, they will match your donation and give it to Microbe TV. They are a 501c3, so your deduction would be federal U.S. tax deductible, and so is Microbe TV as well. So all of your deductions back to April of 2020 are U.S. federal tax free. Of course, it doesn't help you in 2020 because you already filed your taxes for that year. But for 2021, the whole year, you can deduct it from your federal taxes. Dixon Despommier. Oh, and you can go to microbe.tv/contribute for contributing directly to us. Dixon Despommier is at Racaniello.org, thelivingriver.org. Thank you, Dixon.
You're welcome, Vincent, and everybody else. Happy New Year to you all. And see you next year.
One might hope so, yes.
Yes.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun. See you next year.
Alan Dove is at alandove.com. Alan Dove on Twitter. Thanks, Alan.
Thank you. It's always a pleasure.
Brienne Barker is at Drew University BioProf Barker on Twitter. Thanks, Brienne.
Thanks. Great to be here, and Happy New Year.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. And I also want to thank all of you listeners who have contributed over all of the years for your support. How did they say? We couldn't do this without you. It keeps the lights on.
That's right.
If without your support, it would be like this. And if you're listening, I just turned the main light off. I could actually turn both off. So I could turn off the main light and my little highlight, and that's just it there.
Now you look like you're an anonymous source on 2020.
So thank you all for your support, and everyone have a happy New Year.
You too.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next year. Another TWiV is viral.